Our next speaker will be Kohtaro Tanaka, and he'll be telling us about cis-regulatory basis of expression of divergence between recent gene duplicates.
Okay. First, I'd like to thank the organizers for giving me this opportunity to present our ongoing work. And I changed the title a little bit just to include conservation as well. So gene duplication has been recognized as major player in generating the vast number and diversity of genes in our genome for quite some time now. Gene duplication events happen quite frequently. However, most commonly, one of the copies accumulate degenerative mutations and basically become pseudogenized in a matter of a few million years. There are several models that explain how two copies after duplication can be maintained in the genome. So according to classical models, two genes can be maintained in the genome if they diverge both in either in protein function or in tissue-specific expression before they become pseudogenized. So in terms of expression divergence, two copies can split the original expression domain of unduplicated gene, or they can acquire a new expression domain. So they both become essential. Some very recent studies also show that many genes can be retained for over 100 million years in the genome by third type of mechanisms. So according to this third type of model, the duplicates can actually maintain the same tissue specificities but still be kept in the genome. And one way in which this can happen is if two copies rapidly decrease the total expression level to a minimally required level, so they both become essential. And these models further proposes that the relative expression level of each paralog can fluctuate in different lineages by drift. And eventually, when one of the paralogs takes over in terms of tissue expression level in different tissues, then either subfunctionalization or neofunctionalization can ensue after a very long period of time. So what this third model implies is that the cis-regulatory landscape at duplicated loci can continue to be very dynamic evolutionarily, even after the two duplicates become fixed, and even when they don't show significant, drastic expression divergence. But this is something that hasn't been looked before. So in today's talk, we'll simply be looking at the cis-regulatory landscape of a pair of duplicated genes. And I'll be asking these two pretty simple questions. So how conserved or dynamic are the cis-regulatory landscape at duplicated locus? And how these variation correlate with the actual expression variation of the paralogs, including the novel tissue-specific expression. So the two genes we'll be looking at today are CG9336 and 9338, which belong to Ly6 gene superfamily that are present in both insects and vertebrates. The gene products of this superfamily produces a protein which biochemically function by binding to different protein targets and diverse biological contexts. For our two genes, we have yet to be able to characterize the function. So despite the fact that we have generated double null mutants for these two genes, we have yet to be able to observe any phenotype. However, the sequence analysis shows that both of these copies are under purifying selection, so in the wild, they probably have essential functions. In our previous study, looking at the evolution of Ly6 gene family in insects, we found that the two genes have appeared as a result of tandem duplication at the base of Drosophilids. So two copies can be found in all sequenced species of Drosophila, and unduplicated ortholog can still be found in group that are closely related to Drosophilids, such as the two sequence species of tephritid flies, Ceratitis and Bacterocera. So we have also looked at the embryonic tissue-specific expression of the two paralogs. as well as that of the unduplicated ortholog in ceratitis. And we found that tissue expressions such as the ones in peripheral glia and Bolwick's organ have been inherited by both paralogs. On the other hand, expression in tissues such as surface glia and hindgut were only inherited by CG9336. The two paralogs also acquired a novel expression, suggesting potential neofunctionalization. So expression in the heart were acquired by both copies, and expression in the hemocyte was acquired by CG9338 only. So in today's talk, basically, I'll be showing you two main data sets. One will be the expression of these two paralogs, 3.6 and 3.8, in 3 additional species of Drosophila to show the degree of conservation and variation. And then I'll be showing you the cis-regulatory regions controlling this expression. So to look at the expression of the paralogs, we designed riboprobes that are specific to each paralog for the 3 species. The first thing that stood out from this result was that what we considered to be the neofunctionalized domain of the paralogs in Drosophila here were actually not conserved in other species. So the prominent heart expression that we see in the CG9336 of Drosophila melanogaster were progressively more restricted in other species. So this indicated that this heart expression was probably acquired pretty early on after duplication events, but then became spatially modified in lineage-specific manner. As for the hemocyte expression, the only other species besides melanogaster that had this expression was Drosophila ananaceae, suggesting that these acquisitions happened millions of years later after the duplication events. So here are the diagrams summarizing the rest of the results. We found the 3 tissues in which both paralogs were expressed in all species. We found one tissue in the hindgut where CG9336 was expressed in all the species. In surface glia, so on the bottom left, different paralog was expressed depending on the species. So, so to quickly sum up, basically the expression domain of these 2 paralogs seem to have both highly conserved elements and variable ones. So next we determine the cis-regulatory landscape controlling these expression. So to do this, we simply made a GAL4 reporter construct from 5 of the intergenic regions and introns from the CG9336 and 3.8 loci of different species. Then we made the reporter transgenic lines using Drosophila melanogaster and determined which of these regions contain the tissue-specific enhancer activities. So here are just the raw data. And once we determined the enhancer activity of each region, then we combined that data with those of the paralogs expression, and this is what we came up with. So shown in red and pink circles are the genomic regions that contain tissue-specific enhancer activity, not necessarily the exact location of enhancer within these regions. So 2 things stood out from this overall view of these results. First was that many tissues seem to have more than one enhancer, more than one enhancer. So this indicated presence of redundant enhancers for some of these tissues. And another thing that stood out was the location of tissue-specific enhancers seemed to change depending on species. So initially we thought this was pretty exciting, but then 2 recent genome-wide studies came out and have shown that these 2 phenomena are pretty common in the genomes of Drosophila. So what are the correlation between the variation in the cis-regulatory landscape and the paralogous expression? So out of the 3 tissues in which both paralogs were expressed in all species, we found only one of them, the peripheral glia, had the genomic region containing enhanced activity conserved in all species. For the other 2, both location of the enhancer activity as well as the number of regions containing these activities varied. So this suggests that these diverse types of cis-regulatory landscape can drive this conserved gene expression. And for the species that only had one enhancer, basically there is some degree of enhancer sharing going on between the two paralogs. So we had two tissues where only one paralog was expressed. So in these tissues. So in the hindgut, CG9336 was the paralog that was expressed in all species. But despite this conservation, we found that both the number and location of the genomic region containing enhancer activity were variable. And as for the surface glia in which different paralog was expressed depending on the species, we also saw no clear correlation between the location and number of regions containing enhancers. So how about the novel tissue expression? So for the heart, we actually found that the region containing the enhancer activity was conserved in all species, so in the intergenic region between the two paralogs. And moreover, we found that this region from different species drove reporter expression in a spatial pattern similar to those of the native mRNA expression pattern. So this indicated that cis-regulatory difference in these regions are primarily responsible for the interspecific variation in the expression of CG9336 in this particular tissue. And finally, for the hemocyte, we found one location with enhancer activity that was conserved in both melanogaster and ananase. And melanogaster had one additional region containing the enhancer activity. So this indicated that melanogaster acquired secondary hemocyte enhancer more recently. So we found it pretty curious that 2 out of these 3 novel enhancer containing regions mapped to the same genomic regions as the ones that that were highly conserved in different species, because this made us think that maybe these 2 novel expression domains are somehow related to this highly conserved ancestral expression domain, or the enhancers. So to test the idea, we further dissected these 2 genomic regions. So we did this by making a reporter construct with a series of smaller overlapping fragments. And in Drosophila melanogaster, we found that the peripheral glia and the heart enhancer activity was localized to a relatively small region of 418 base pair, and the peripheral glia and hemocyte enhancer activity to 541 base pair regions. And when we did this in other species with somewhat cruder resolution, we found that these 2 activities always were found to be in similar or in a close location. So this suggested to us that the heart enhancer and the first hemocyte enhancer probably arose once near the ancient glial enhancer and then was conserved since then. And it also suggests the possibility that the emergence of these 2 novel enhancers may have occurred by modification of the ancient glial enhancer activity. So this is something we are testing now by dissecting these regions even further. We also found that, so we also narrowed down the enhancer activity of the second hemocyte region in Drosophila melanogaster to a small piece of 165 base pair region. So right now we are comparing this region to the orthologous regions from more closely related species to determine the exact timing of the emergence of this enhancer. So just to summarize, I've shown you that expression of fixed paralogs and the underlying cis-regulatory landscape can be very variable in different lineages. Our 2 paralogs maintain some ancestral expression domain from the single ortholog despite changes in underlying cis-regulatory landscape. The novel expression domains seem to be acquired both immediately following duplication events and more recently in a lineage-specific manner. And this acquisition may be due to novel enhancers emerging through potential modification of a pre-existing enhancer. So with this, I'd like to acknowledge members of the Susanna lab, especially the two technicians who generated a lot of these data, and Elio Susanna, the boss, and other people, and our funding source. Thank you very much for your attention.
Thank you.